This is a production of Cornell University.
Thank you all for being here. I mean, this is quite an experience, even setting up this thing. Thank you, David, for the nice introduction and for the invitation. And I'm not sure if Adam's in the audience, but thank you for the initial invitation and for organizing my visit. And I would like to also thank some of my previous professors for showing up today, like Tom. You were on my A exam that David brought up. It was a nice experience. You know, I had really dreaded it, but you know, all the professors were very nice. And it's really nice to be back here. It's not easy to get to Ithaca, but it's really nice to be here. Yeah, so today I would like to tell you about a new research direction. Sorry, I changed the title. I forgot about the title that I gave like a year and a half ago. So today I would like to first give a very brief introduction, a brief overview of our work on microRNAs, and then go on to talk about some of the new research directions. So my experience in David's lab really instilled in me appreciation for RNA, not only as an information carrier, but also as the nexus of gene expression mechanism. And you know, now you all know that many processes occur at the RNA level, and these processes then bear on the translatability, stability, and maybe the subcellular localization of RNAs. And thus these processes impact gene expression. Oh, I guess I need to move this thing, huh? Oh, yeah. Yeah, thank you, Maureen. Do I turn this off?
Yeah, top left of that box. Sorry, right hand side. Double dash, all the way over. Those people are okay.
Nice.
Oh,
I see.
If you touch that little bar right there.
Oh, yeah. Thank you. So when, you know, my experience in David's lab really, you know, made me appreciate the power of RNA. Then I did my postdoc with Elliot Meyer, who was working on flower development. And I had to say for like 4 years, RNA did not come up. So all of the players in flower development at the time were transcription factors. And I was very interested in genes that, oops, now it's not advancing. So I was really interested in genes that pattern the reproductive organs in the flower. So these reproductive organs in the center. The only gene known at the time was A-gammas. You can see that in this mutant, the flower doesn't have reproductive organs. The reproductive organs are transformed into petals and sepals. So what I did was to take advantage of a weak A-gammas allele at the time that had still these male reproductive organs, the stamens. And I did the mutagenesis screen, the EMS screen, to look for genes that help pattern the reproductive organs. And this screen led me to 2 genes, which I named FA1 and FA2. And these 2 genes basically landed me my first position. And in my own group, we decided to take advantage of this double mutant FA1, FA2 to conduct another genetic screen to look for more genes that pattern the reproductive organs in the flower. So what we did was to mutagenize this and look for enhancers that would enhance the floral homeotic phenotype. As you can see, in some of these mutants, the stamens are transformed to petals. And most interestingly, when we cloned these genes, we found that many of them actually are players in RNA metabolism. So this brought us back to the RNA world, particularly this gene called HEN1. So when we cloned this gene, we realized that the protein is a so-called pioneer protein with unknown molecular function at the time. But it does these animal homologs, even some homologs in bacterial species. And it was the HEN1 single mutant phenotype that gave us the clues to the molecular function of this protein. At the time, another postdoc in Elliott's lab, Steve Jacobson, was working on a mutant called CAF. And these two genotypes, I realized, are so similar. And CAF turned out to be dicer-like-1, one of the dicer-like proteins in Arabidopsis. This made me realize that maybe both genes actually act in small RNA metabolism in plants. So at the time, there were no known small RNAs. So we went about cloning the first few microRNAs from plants, and then we looked to see whether these small RNAs are gone or reduced in abundance in the HEN1 or the CAF mutant, and that was the case. So this led us right back to the RNA world. And then we went on to show that the reason HEN1 mutates to the floral meiotic phenotype that we saw is most likely because of this microRNA. So it turns out that this microRNA is a repressor of AP2, which is a very important gene in floral, in specifying the petal identity in flowers. So that was in the early 2000s. And we decided to make a switch from studying flower development to studying RNAs, because RNAs were just known at the time and very little were just isolated at the time. Very little was known about them. So one of the first questions that we addressed was how microRNAs are made. So we, using molecular genetics, found Pol2, mediator, and dicer-like-1 are required for microRNA biogenesis. And it's very easy to put these proteins into this pathway just based on their known molecular functions. But we really scratched our heads on the molecular function of HEN1. So what does this gene do? And it turns out that we later found out through biochemical studies that HEN1 actually is a small methyltransferase. It works on dicer product. It's not sequence specific. And it puts a methyl' onto the 2' OH of the 3' terminal ribose. And we then went on to show that these two major types of small RNAs, plants, microRNAs and siRNAs, they all carry the 3' end. And because HEN1 has these animal homologs, we also looked into whether animal microRNAs had this methyl group. And it turned out that very few of them actually do. And at the time, we didn't find any. But it was very gratifying to see that many labs showed that pi RNAs in animals are methylated, and they're methylated by HEN1 homologs. So then we looked into what this methylation leads to microRNAs, and we found that in the HEN1 mutant, what happens is that these microRNAs become 3' truncated or 3' uridylated. And at the time when we published this work in Current Biology, Guy Riddle, a editor for the journal Science, picked up our paper and highlighted it. And I really like this title that he gave to his article. So to me instead of you. Later on, others also showed that in terms of pi RNAs, you have the same phenotype basically. So in C. elegans, Tetrahymena, Drosophila, HEN1 mutants, what happens is that these pi RNAs become 3' truncated and 3' uridylated. So this is a conserved process in small RNA metabolism. So then this led us to pursue the second big question in small RNA biology, and that is how microRNAs are degraded. We presume that these processes lead to microRNA degradation. So we looked for the enzymes that cause 3' truncation of microRNAs and 3' uridylation of microRNAs. So we presume that would be an exonuclease, this would be a nucleotidotransferase. Looked for them using reverse genetics approaches, and we identified a family of small RNA degrading nucleases, and we identified 2 nucleotidotransferases that can do this uridylation. And by doing biochemistry, we showed that this methyl group really prevents the activities of these nucleotidotransferases, and it deters but does not completely inhibit SDN enzymes. More importantly, we were able to show that these enzymes can act on Argonaute-bound small RNAs. Remember, the small RNAs in vivo, they're bound by Argonaute proteins. And these enzymes can really work on Argonaute-bound proteins in vitro, suggesting that they do play a role in regulating small RNA abundance in vivo. And I would say that related to our microRNA degradation, there is a result that I would like to highlight. That is STTM. So STTM stands for short tandem target mimic. This was a technology that was developed by collaborator Guiliang Tang at Michigan Tech University. So STTM is basically a 90 nucleotide or so RNA that can be expressed with any promoter that you want in vivo. And this RNA consists of microRNA binding sites separated per a linker. And microRNA binding site is such that when it pairs with a microRNA, there would be a bulge in the middle. And this bulge prevents the microRNA from causing cleavage of the STTM RNA. So the idea from the Tang lab was that they would express these STTM RNAs in vivo to soak up the cognate microRNA to prevent the microRNA from working on its own targets. But when they introduced the STTM constructs into plants, they saw that they led to the reduction of the amount of the cognate microRNA, very specifically. So we suspected that this may be due to microRNA degradation. So we collaborated to show that the STTM RNA was actually causing the degradation of the cognate microRNA through the SDN exonucleases. And this led us to the idea that maybe we could develop a resource for studying microRNA function in vivo. So what we did was to come up with a collection of conserved STTM constructs for conserved as well as sequence-specific microRNAs. And we introduced them into rice, maize, soybean, tomato, and Arabidopsis. And the STTM technology basically works in all these plants. And you can see here that you can get specific phenotypic traits by inhibiting specific. And these constructs are now available through AdGene. So next, I would like to go on to talk about some of the ongoing research. And I will very briefly talk about our current research on non-cell autonomous microRNAs, and then I'll mainly focus on this new direction. So microRNAs were known starting from the year 2000. And in the early years, people thought that microRNAs act cell autonomously. And they're so important in patterning, so you would expect them to act cell autonomously. However, in plants at least, there has been increasing evidence that microRNAs can move from cell to cell and use systemically. And in some circumstances, microRNAs have been shown to serve as signals in developmental patterning. For example, in this case, this is a diagram of the Arabidopsis root. This particular microRNA is only expressed in the endodermis, but it forms an activity gradient towards the center of the root. And this is transformed into the reverse gradient of its target gene. And the different levels of expression of the target gene would lead to different cell fates in the vasculature. So in essence, this microRNA is functioning as a morphogen in development. And also in leaf pattern, there are two small RNA gradients. One is a siRNA, the other is the same microRNA. Here, these two gradients help to specify the two sides of leaves. And I really like this study. This is a recently published study earlier this year from Jianxin Ma's lab. What they did was to do a soybean, common bean heavy graft. And they were able to use the SNPs present in these two species to identify species-specific small RNAs. And by doing so, they found basically movement from shoot to root of a lot of small RNAs, endogenous siRNAs as well as microRNAs. So they're moving en masse. Whether they all serve as signals, we don't know. But at least, there are examples of small RNAs serving as signals in development and in stress responses in plants. And we realized that very little is known about how, what are the processes that regulate the cell-to-cell movement of small RNAs. So we decided to do our genetic screen to look for mutants that are compromised in the cell-to-cell movement of microRNAs. So we took advantage of this line This is a artificial, this line expresses an artificial microRNA using a vasculature specific promoter. But this microRNA moves out of the site of synthesis into leaf mesophyll cells. So that's where you see this vasculature-centered leaf bleaching, because the bit of the artificial microRNA is important for chlorophyll biosynthesis. So then we mutagenize this and we look for mutants that have reduced area of leaf bleaching. So hopefully some of those mutants would be in genes that are important to regulate the cell-to-cell movement of microRNAs. Of course, this screen is specific to microRNA mobility. We could get microRNA biogenesis mutants, for example. So they would show reduced artificial microRNA as well as levels of endogenous microRNAs. And we could screen these out by looking at this. So we keep the ones that have normal levels of the artificial microRNA and normal levels of endogenous microRNAs, either be in microRNA activity or microRNA movement. So then we do further experiments to tease them out. So right now we're working with a few of these options, and we have some interesting results, but I think today I will instead talk about another research direction. So you could stay tuned for work in this area. So today I would like to talk mainly about our recent work on RNA NAD capping. So you all know that eukaryotic mRNAs carry a M7G cap. So this is a methylguanosine attached to the first nucleotide of the RNA through a 5' to 5' triphosphate linkage. And this cap obviously is very important for RNA metabolic processes, but in recent years, alternative caps have emerged. And these are really interesting molecules. They're basically cellular metabolites. I think the sheer fact that these cellular metabolites could serve as mRNA caps suggests that cellular metabolism could intersect with RNA metabolism. And today I'm going to basically refer NAD plus and NADH caps as the RNA NAD cap. It can be distinguished, but the profiling approaches that I'm going to talk about cannot effectively distinguish the two. And also, please metabolites, they are all ATP analogs. And this is important when it comes to how these caps are deposited onto RNA. Because they are ATP analogs, they can be incorporated into RNA by RNA polymerase as the initiating nucleotide. So they're used in place of ATP. In fact, it was shown in vitro that bacterial RNA polymerases, mitochondrial RNA polymerase, and yeast Pol2 can all use NAD in vitro as the initiating nucleotide. In fact, structural studies using bacterial RNA polymerase and the DNA template strand in red and the NAD-RNA showed that the NAD moiety is actually well coordinated by the active center amino acids in the RNA polymerase. Because of the co-transcriptional deposition in bacteria, it has been shown that NAD-RNA production, the efficiency of NAD-RNA capping is related to the promoter sequences. And in human, there's some evidence that there may be post-transcriptional NAD capping as well, but the mechanism is unknown. So the NAD cap was first found by David Liu's group in 2009. So what they did was to take bacterial RNA, remove the small molecule fraction, and then digest the macromolecule fraction by nuclease P1, so that now you have nucleotides. And then they subjected the nucleotides to LC-MS analysis, and they found NAD. And further analysis showed that the NAD was actually at the very 5' end of the RNA. So we developed a similar LC-MS approach to see whether Arabidopsis RNA contains NAD. And indeed, we were able to find NAD from both total RNA and mRNA. And if you look at the amount of NAD in total RNA versus mRNA, you can conclude that NAD is primarily in mRNA. So this approach tells you that there is NAD in RNA, but it doesn't tell you which RNA species contains the NAD cap. And this was the development of a revolutionary approach to sequence NAD-capped RNAs that was developed in 2015. So in this NAD capture-seq, what they did was to convert first the NAD-RNA into an alkyne RNA using an enzyme ADPRC and the alkyne. And then the second step would be a click chemistry reaction where the alkyne RNA reacts with the biotin azide to generate phenylated RNA. And then the biotinylated RNA can be purified and sequenced. So using this approach, it was found that bacteria, yeast, and human cells contain NAD-capped RNAs. So I will refer to NAD-capped RNAs as NAD-RNA from now on. And using a similar approach, my lab and Brian Gregory's lab showed that plants also have NAD-RNAs. So basically, this was the same NAD-RNA capturing approach that I showed in the previous slide. I just wanted to use this slide to point out that when we do the seq, we also include ADPRC minus samples where we leave out this enzyme during the capturing. So these would be the RNAs that got non-specific attached to the beads. And we did the sequencing with Arabidopsis inflorescences and seedlings in replicates. And then by comparing the amount of the read count of the ADPRC plus as compared to the ADPRC minus samples, we were able to identify thousands of genes that produce NAD-RNAs. And there was a good overlap between the two types of tissues. So where are these NAD-RNAs from? We found that they're mainly from the nuclear genome. And if you take a look at the browser views, it's very easy to see that these NAD-RNAs are properly spliced. So here are the ADPRC plus tracks and the ADPRC minus tracks, which are very clean. And you can see that the introns are removed. And most NAD-RNAs are also polyadenylated. So here we did NAD-RNA capture followed by RT-qPCR with either random primers or the oligo-DT primer. And you can see that we get the same amount of NAD-RNAs for these genes. And we also looked into this at the genomic scale. So we first isolated poly-A RNA, and then we did NAD capture-seq and compared that to total RNA. And you can see that here we're plotting the ADPRC plus minus ratio to represent the levels of NAD-RNAs. And you can see a very good concordance. And here I wanted to also highlight NAD tag-seq from my collaborator, E.G. Xia from Hong Kong Baptist University. So I thought this was a very innovative approach. They converted NAD-RNA basically eventually to a chimeric RNA containing this tag RNA. So the first step in the capture is the same. You convert NAD-RNA to this alkyne RNA. And then the second step, instead of using biotin azide, they used azide covalently linked to this tag RNA. So then in the end, you end up getting this chimeric RNA. And then they could subject the library to nanopore single molecule sequencing. So in nanopore single molecule sequencing, you start from the 3' end of the RNA. So when you sequence and you get the tag RNA sequence, then you know that the original RNA was NAD-RNA. So advantages to NAD tag-seq. One is that you can actually derive the ratio of NAD-RNA versus non-NAD-RNA. So if you think about two types of RNAs from any gene, NAD-RNA and M7G RNA. After the tagging, the NAD-RNA contains the tag RNA, but the M7G RNA does not. Then you can either capture the NAD-RNA through a oligonucleotide, DNA oligonucleotide that's complementary to the tag RNA, or you can just directly sequence this mixture. If you directly sequence this mixture, then obviously you could get the ratios of the NAD-RNA and M7G RNA. So when they did this, they estimated that NAD-RNA is about 5% of M7G RNA. But I will tell you that this is a huge underestimate when it comes to some genes later on. And another advantage of this NAD tag-seq is that you can get full-length sure that these NAD-RNAs are spliced and polyadenylated. And interestingly, they found that the NAD-RNAs have the same 3' UTR, but shorter 5' UTR compared to M7G RNAs. So despite having short 5' UTR, the NAD-RNAs appear to have the same coding potential as M7G RNAs. So then we wondered whether NAD-RNAs are translated. This is not a very easy question to address, so we turn to a simpler question, that is whether NAD-RNAs are polysome associated. So we got Arabidopsis polysomes, and we isolated the polysomal RNA, and then we subjected the polysomal RNA to a NAD capture-seq. And we used total RNA isolated at the same time for comparison. And here I'm plotting here by comparing ADPRC plus minus, you can derive the NAD RNAs, and you can see that we see a lot of NAD RNAs on polysomes. Here I'm plotting the ADPRC plus minus ratio to represent the levels of NAD RNAs. And you can see that in the polysomal fraction, we actually see higher ADPRC plus minus ratio, suggesting that we see preferential polysome association for NAD RNAs. So are they actually associated with translating ribosomes? So for this, we turn to the treatment with pyrimycin. So pyrimycin splits translating ribosomes to ribosomal subunits. And you can see that the treatment works. The polysomal fraction is reduced upon treatment. Monosomal fraction is increased. So then we isolated RNA from these different fractions, and we performed NAD RNA capture and then real-time RT-PCR. So here, basically, we're looking at mock-treated samples. You can see this significant enrichment of the NAD RNA in the polysomal fraction as compared to the non-polysomal fraction. But upon pyrimycin treatment, this is reduced. You don't see that enrichment anymore, or you even see the movement of the NAD RNA into the non-polysomal fraction. So this suggests that NAD RNAs are associated with translating ribosomes. So during our efforts in NAD RNA profiling, we realized some issues with NAD capture-seq, and we sought to develop new methods to profile NAD RNAs. And I'm going to talk about this new SPAAC NAD-seq. So we looked at our NAD capture-seq read coverage along genes. So this is the gene body coverage of reads. And we see this 5' skew in the ADPRC plus samples. And we usually don't see that with RNA-seq or with the ADPRC minus samples, which are shown by the dotted lines. And we thought about this and we realized that that's because of copper. So in the NAD capturing scheme, the first step is to turn NAD RNA into alkyne RNA. And the second step uses this copper catalyzed click chemistry reaction to turn alkyne RNA into biotin RNA. But copper ions are known to degrade RNA, to fragment RNA. So when you have RNA fragmentation together with capturing of the 5' end of the RNA, you would have this 5' skew in the counts, in the read coverage. Also, what this means is that you will have a lot of RNA degradation going on during your converting NAD RNA to biotin RNA. So we decided to improve upon NAD capture-seq. So this is the click chemistry. It's called copper catalyzed alkyne azide cycloadditi reaction. I will refer to this as the SPAAC reaction. So here, what we do is to use ADPRC to basically convert the NAD RNA to this alkyne RNA, and then have this click chemistry chemical reaction to generate biotin RNA. And there is a second popular click chemistry reaction called SPAAC, strain-promoted azide-alkyne cycloadditi reaction. So here, my understanding is that the alkyne being in cyclic form, ring form, there's strain in it, and the strain would allow the reaction to occur in the absence of any catalyst. So we thought that we should adopt this click chemistry reaction to do an NAD-RNA capture. So remember in the previous reaction, they appended NAD-RNA to this alkyne, but this alkyne is very bulky in this SPAAC reaction. So we thought that maybe we should try to append the RNA to the azide using ADPRC. So can we do that? We need to first show that this works. So if this works, then we would use a biotin tagged alkyne to generate biotinylated RNA. So we looked into this by first reacting NAD and alkyne or NAD with azide in the presence of ADPRC. And then we would look at the product using LCMS to show that indeed we have the right product. So indeed, that was the case. So ADPRC could catalyze the reaction between NAD and the azide. So then we tested this with Arabidopsis RNA. So what we do here is we take either total RNA or mRNA. We perform these reactions to turn the RNA into biotin RNA. And then we run the RNA in the gel, and then we detect the RNA using streptavidin HRP. So the signals that you see here represent biotinylated RNA. So let's first take a total RNA. So you can see that with the SPAAC reaction, the sizes of the RNA are much larger than those with the Kuak reaction. This is consistent with the lack of RNA degradation in the SPAAC reaction. But you may also notice that in the negative controls, so even without ADPRC, we still see signals, and these are mainly ribosomal RNA. And this is because ribosomal RNAs perhaps have some modifications that could also lead to the biotin conversion. This is not a problem with mRNA. So with the Arabidopsis mRNA, the negative control lanes are very clean. And you can see that with the SPAAC reaction, we got much, much stronger signals as compared to the Kuak reaction. So we were very excited when we saw this result, but our excitement lasted maybe 5 minutes. Then we realized that could it be that we are having an issue? Maybe what we thought we were doing was to convert NAD-RNA to biotin RNA, but maybe M7G RNAs were also reacting. That'd be a problem. And I should mention that NAD captureSeq was initially developed for E. coli RNA, but it was adapted to eukaryotic RNA without extensive quality control to see whether M7G RNAs also react. So we decided to look into this. So if M7G RNA can react, so we took M7G PPA to represent M7G RNA, and then we performed the reaction with ADPRC and the azide. And we would expect if the reaction works, then we would get M7G, which we could detect by LCMS. And indeed, we were able to detect M7G to our disappointment. And this actually happens no matter whether you use the azide or the alkyne in the reaction. So basically, the original NAD captureSeq had the same issue. But what's really nice is that these peaks are pretty small. As you can see, it's very close to the baseline, suggesting that this is not a very efficient reaction. So we wanted to look to the efficiencies of NAD RNA capture or the efficiencies of RNA biotinylation for NAD RNA and M7G RNA. So what we did was to in vitro transcribe a NAD RNA and M7G RNA of different sizes. Then we mixed the two at different molar ratios, and then we conducted the reactions. And you can see that indeed, at this higher M7G RNA ratio, we could see that the M7G RNA is converted to biotin RNA. But luckily, the efficiency is much lower than for NAD RNA. And we suspect that this was also going on even for the Kuak reaction, but maybe because of RNA degradation, we cannot clearly assign a bend to the M7G RNA. So then we thought that we should try to remove M7G RNAs in the before sequencing. So we tried to see whether this commercially available anti-M7G antibody would work. So what we did was to perform in vitro transcription, and then we mix 100 nanogram of NAD-RNA with 10 micrograms of M7G RNA in vitro. And then we either not treat the samples or treat the samples with M7G beads. Afterwards, we would perform the SPAAC reaction to detect RNA. So without any treatment, you would see the NAD-RNA and the M7G RNA being biotinylated. But after the M7G treatment, you do not see the M7G RNA signal anymore. So we were able to remove the M7G RNA. So next, we tried this with Arabidopsis RNA. So we took Arabidopsis mRNA and then did either mock treatment or M7G treatment. And then we digested the RNA into nucleotides. And we look at M7G PPPA as a readout for M7G RNA and NAD as a readout for NAD RNA. So basically in the flow through, we see about 80% reduction of the M7G RNA. And we do not see any reduction in the NAD RNA by this M7G antibody. So according to our calculations, this should take care of the problem of M7G RNA interfering with or contaminating the NAD RNA profiling. So basically now we have developed this SPAAC NAD using the SPAAC reaction and also combining this with M7G RNA depletion. And you could do that with either the SPAAC reaction or the Kuak reaction. And we were able to show that the SPAAC reaction gave higher NAD-RNA signal as well as better gene body coverage. So this is with the Kuak reaction, you see the 5' skew, but with the SPAAC reaction, you have better gene body coverage. And you can see that better for individual genes as well. This is the Kuak reaction, this is the SPAAC reaction, and this is the regular RNA-seq. So from developed SPAAC NAD-seq with M7G RNA depletion, we got some gene, go terms for the genes that produce NAD-RNAs. And a lot of these are so-called response genes. So these are involved in environmental responses. And the original NAD captureSeq also identified a lot of the response-related go terms, except for this go term that is photosynthesis. We now think that this is probably not, these are probably not really NAD-RNAs. The photosynthetic genes are expressed at very high levels. And I think in this case, these genes showed up probably because the M7G RNAs came up in the capture scheme. And my collaborator also upgraded their NAD tag-seq to NAD tag-seq 2 using the SPAAC approach. And here they applied NAD tag-seq 2 to bacterial RNAs. And I wanted to make two points here. One is that they looked at the stationary phase versus the exponential phase. And they were able to show that for the same genes, the amount of NAD-RNA differs between the two growth phases, suggesting that NAD-RNA production is regulated. And another point that I wanted to make is that for some genes, the percentage of NAD-RNAs in the total transcript pool can be very high. It can be up to 70%. So for these genes, we presume that the NAD-RNA capping plays some roles in regulating the expression. So then the question is, what is the molecular or biological function of RNA NAD capping? I think we're still in the early days. I think there's still a lot to be done, but there are some conclusions that can be made. For example, in E. coli, it was shown that the NAD cap stabilizes RNA. The RNA with the NAD cap is more stable than the RNA without the NAD cap. But eukaryotes is the opposite. So in a human cell line, for example, NAD-RNA, when the NAD-RNA was transfected into the cell line, it had shorter half-life compared to M7G RNA. And the half-life depends on an enzyme called DXO, which I will talk about next. And here, I also wanted to mention that in this particular study, they transfected M7G luciferase RNA and NAD luciferase RNA into the human cell line. And they did not see any luciferase being made from the NAD RNA. So it seems like, at least from this study, the NAD cap does not support translation. But remember, I was telling you that in Arabidopsis, NAD RNAs are found on polysomes. So I think this aspect of things is still controversial. We still need to figure out whether in vivo NAD RNAs are translated. So in terms of functions of NAD RNAs, I think studying the NAD decoding enzymes or denadding enzymes would be informative too. So there are actually two major classes of NAD decapping enzymes. One is called DXO. So DXO actually takes out the entire NAD moiety from the RNA, leaving a 5' phosphate RNA. And the DXO mutant has very severe phenotypes. So DXO actually has two activities. First is this denadding activity. Second is that it has 5' to 3' exonuclease activity that acts on 5' monophosphate RNA. So presumably, DXO could degrade NAD RNA by first decapping and then degrading the rest of the RNA. In fact, my collaborator E.G. Xia's lab showed that when they transfected a NAD-capped luciferase RNA into wild-type cells, they see very small, very fast degradation of the RNA. But the half-life is longer in the DXO mutant protoplast, suggesting that indeed in protoplasts, DXO helps to degrade NAD RNA. So how about in vivo implanta? Work from Brian Gregory and Joanna Kufo's lab showed very interestingly that in the absence of DXO, many genes actually now produce small RNAs, endogenous siRNAs. So the idea is that in the absence of DXO, RNAi actually is used as a backup to degrade these RNAs. So in fact, this is a major RNAi enzyme in plants. And in fact, when you make a double mutant between DCL4 and DXO, you get very, very severe phenotypes, seedling lethality, basically. So it seems like DXO is to degrade some NAD RNAs, but in the absence of DXO, RNAi can help to some extent. However, I think here there's still a lot to be done. If you look at a DXO mutant that's transformed with a catalytic mutant you see pretty much good rescue of the phenotype. So whether in vivo DXO really has this denadding activity or whether the denadding activity is really relevant to the phenotype, we don't know yet. So a second class of NAD-RNA decapping enzyme is represented in E. coli NUD-C. So this is a Nudix domain protein. And so the NUD-C actually has a different activity from DXO, so it removes the NMN nicotinamide moiety of the NAD RNA, again leaving a 5' monophosphate RNA. So if you look at the Nudix family, this is a pretty family in Arabidopsis of 29 or so members. And in fact, the famous DCP2, which is the M7G decapping enzyme, is a Nudix family protein. So we looked for NUDT homolog or ortholog in Arabidopsis, and it appears to be NUDT19. And I'm not sure if I have the time, maybe not. So I won't show you the enzymatic activity assays that we did, but we showed that indeed it has NAD-RNA decapping activity in vitro. So then what are the phenotypes of NADT19 mutants? So there is apparently a null mutant in which there's no protein that could be detected, and this mutant looks very normal. However, upon stress, so these knockdown and knockout mutants, they seem to be more tolerant to stresses. So I think maybe NADT19 are more response related in terms of development related. So I think I will stop here and summarize what I told you. So basically, we found that NAD-RNAs are widespread in the transcriptome, and it appears that in vivo NAD-RNAs are less stable than M7G RNAs. And we found that NAD-RNAs are associated with translating ribosomes. And we have identified these two decapping enzymes. And I think studying these decapping enzymes would lead to understanding of the molecular and biological functions of NAD-RNAs. So these are some of the questions that we will be addressing in the near future. Are these NAD-RNAs really translated? And what are the biological processes regulated by RNA and NAD capping? And I'd like to thank the people involved in this work. Most importantly, EGCA, we have been collaborating in the past 3 to 5 years on developing this new sequencing procedure and on the decapping assays. And Guiliang Tang, I mentioned, developed the STTM technology. We are collaborating with Karen Brown to look at the translation of NAD-RNAs in the wheat germ system. And these are a former graduate student and 2 current postdocs who contributed to this work. And I would like to thank the UCR Metabolomics core facility as well. The LCMS was done there. Okay, well, thank you for your attention. And thank you guys also who participated via Zoom. I'm not sure whether the Zoom questions can be monitored.
Yeah, I want to give first shot to people in the room. Can I get in the back row there?
Andrew?
Yeah, wondering if you know that the CAC or NAD binding complex, right? For the more general purposes, have you looked at the translation initiation factors?
Yeah, very good questions. So Andrew was asking whether we looked at proteins that bind the NAD cap and whether the translation initiation factors bind them. So we did in vitro. So we incubated NAD-RNA and M7G RNA with extracts, did the pull down and mass spec analysis. And of course, these analyses, you get a lot of proteins. So we're still combing through them. So then it's possible that we could identify some readers of NAD this way. In terms of the initiation factors, we looked at them individually. For example, EF4E and EIF2. They're very sticky, so they bind in vitro to not only M7G RNA and NAD RNA, they also bind 5' PPP RNA. So we haven't been able to get the assays to work yet.
Over there.
Yeah, I guess you talked a lot about the decapping enzymes at the end, but is there like a pathway for the capping enzyme?
So the capping, so far, it's thought to be done by RNA polymerase. Okay. So RNA polymerase uses NAD as the initiating nucleotide. And this is studied much better in prokaryotes. In E. coli, they were able to show that the promoter, the transcription start sites really matter. For example, the first nucleotide has to be A, because NAD is an ATP analog. And the -1 position is really important, as well as the +1 position. The NMN moiety actually can pair with the DNA non-template strand. I'm sure there are features that determine NAD capping efficiency.
Can you read the question from the chat?
I cannot see this very well, my old eyes. So
this
is better.
Yeah. Or I can read it.
Whether my microRNA targets. you know, whether microRNA targets are preferentially NAD capped. We haven't actually looked into that. Yeah, we don't know. Yeah. Tom?
So you showed us this interesting result where catalytically inactive protein is complementing the mutant. Could be very interesting, but is the mutant for sure a null? You have a partial deformed protein that is like an intragenic complementation instead of something mechanistic.
Intragenic complementation.
DNA 1-2 or something.
Oh, whether maybe the catalytic mutant version was complexing, for example, with the DXO1-2 protein. Oh, that's a possibility. Yeah, right. Yeah, this goes back. Right. No. I mean, it's a catalytic null for sure. But the DXO1-2, I think it's a it has a tDNA insertion somewhere, but I don't know whether it's a complete null. Yeah, great. This goes back to your East genetics course.
It's a very nice thought, and particularly by the enzyme is more tolerant to stress, and it kind of reminds me that NAD and NAD actually involved in cellular resolution. This is like kind of it related to and it's also related to energy. And you also mentioned that the NAD capped RNAs are more enriched in the electrons. So it's possible, like the cell is able to use the NAD capped RNAs as a sensor for its energetic synthesis, and then accordingly regulates the translation of some of these RNAs as an output. Like I don't know if I expressed it clearly. Like it's,
yeah, so I think so. I think the question is whether, because NAD is a redox and energy agent, so could it be that NAD-RNAs are used as a sensor for the redox state, for example, energy state of the cell? I think it's possible. In fact, Brian Gregory's lab showed that in DXO mutants, for example, a lot of the NAD metabolic enzymes, these genes are induced. So there seems to be crosstalk with NAD metabolism. The NAD-RNA metabolism may have crosstalk with the cellular metabolite metabolism. Yeah.
Earlier question, I got a little confused here. Maybe with your diagram about the directionality of the NAD cap. Is it a 5' or 3' direction?
Yes. You mean the place, the position of the NAD?
Yeah.
Yeah. So this is the NAD, and then this is the first, the second nucleotide of it. So the RNA would go like that. So this is the first position. Yeah, so this is the NMN moiety, and then this is the A.
Oh, yeah. Okay, so it's normal, like.
Yeah, the rest of the RNA is just RNA. Yeah.
Okay.
Over here, please.
Eric, yeah.
So aside from photosynthesis, which you argued was perhaps an artifact, what other geontology terms are overworked statistically for any RNAs?
So these are, the first one is actually response to cadmium ion. So we actually are looking into cadmium sensitivity of these decapping enzyme mutants. Yeah, so that's something we're looking into. And then there's also response to bacteria, response to light. There's a lot of response-related genes. Yeah. Margaret.
So I think you're treating the RNA
Mm-hmm.
bacteria or dynamic. Are you looking into like dynamic capacity over protein?
We actually have not. That's Yeah, it's hard to do because the capturing approach, the sequencing, you cannot work with a lot of samples at the same time. But the NUDG mutant has a circadian defect. Yeah.
Can I get one question? Because you already have one. So you showed all these polysome association. It seemed pretty convincing on a whole genome level that these RNAs were ribosome associated, and yet you balance that against like one luciferase transfection experiment in human cells or something. So what other explanation is there for the polysome results that you got?
Well, one caveat is that that was done with NAD captureSeq, where we did not do M7G RNA depletion. So we wonder whether maybe M7G RNAs were causing the problem or being actually captured there. So we actually evaluated that. So we we actually took basically here the blue genes in the preferentially translated genes, and the green ones are polysome deleted genes. So we actually took these two groups and looked at their NAD-RNA levels, and we don't really see a difference. So this would argue against M7G RNAs contributing to this. But still, I think we now need to go back to do M7G RNA depletion and then the SPAAC reaction. So we'll see. Andrew?
I think you said that the 5-part PR is shorter. So I got that right. Does that mean that the capture are initiated with it?
Yeah, so that's the idea for so that was from NAD-TAG-seq, so from nanopore single molecule sequencing. However, because remember in the capturing scheme, you have this bulky chunk of stuff between the RNA and the tag RNA. So because of that, the nanopore cannot call precisely the site of transcription initiation. So you can only say maybe it's within these 10 nucleotides or so. And at that precision, at that level, we could definitely say that M7G RNAs have longer 5' UTRs, but we cannot say where is the initiating site for NAD RNA. Right, for a lot of the genes, yes, yeah.
One quick question. Because you see a lot of response with genes getting enriched in your mRNA. I was wondering whether it is possible that those genes have H2AZ enriched.
Oh, yeah.
H2AZ
Yeah. The question is whether these NAD capped RNAs are from genes that are H2AZ enriched, because H2AZ is this variant that's associated with response or stimulus response related genes. And we haven't looked into that, but yeah, that could be something into, yeah.
Yeah, I think in the interest of time, and thanks for,
yeah,
we know you lost a lot of time to technology, but thanks for bearing with it. And let's thank Xuemei again for, it was exciting.
Thank you. Yeah, thank you all for being here in person. It's great to see you.
This has been a production of Cornell University, on the web at cornell.edu.